National Chiao Tung University has developed the world's first quantitative COVID-19 rapid screening detection system, which can test for COVID in just 5 minutes.
Unlike the traditional methods of qualitative screening, which can only detect whether someone has been infected, this system can also detect the level of coronavirus antibodies, antigens, and nucleic acids in the human body.
It is therefore able to analyze the stage and level of COVID-19 infection of the patient. Let's see it in action.
Chiao Tung University has developed the world's first quantitative rapid screening detection system. Not only can it detect whether someone has COVID-19, it can also determine the level of infection of the patient. Drop a 0.02 millimeters blood sample into this machine, which is equipped with a biological sensor chip and a chemical sensor device. In just 5 minutes, it can identify the level of coronavirus antibodies, antigens, and nucleic acids in the body. It's the world's first quantitative rapid screening system for COVID-19.
In our test, we just using the antibody and antigen and nucleus to specific target the virus. So we can quantitatively the virus, the concentration of the virus and the concentration of the antibody. And using this technology, we can distinguish which step of the patients.
我们可以不是只有单一个晶片组的部分，我们可以是 multiple 的。
This quantitative detection system was developed by National Chiao Tung University in collaboration with biomedical companies. Unlike qualitative screening, which can only obtain whether someone has been infected, the screening system delivers data on the level of antibodies, antigens, and nucleic acids of the novel coronavirus in the human body. Based on the concentration of biomolecules, it can judge the level and stage of the infection and formulate follow-up isolation and treatment methods to provide the best medical care for the patient.
Traditional test right now is rapid test because if right now rapid test a yes and no, they don't know how many virus or how many concentration of the antibodies. So that is the difference because if we have a no the virus particle or the antibody concentrations, we can distinguish the different stage of the patients. The accuracy right now, we just using the clinical samples, 30 positive sample and 70, 90 negative samples. We can all distinguish all of them. So after so using the clinical sample right now, we say so 100%.
Chiao Tung University says it is currently applying for certification from Taiwan's Food and Drug Administration. The screening system has already obtained emergency approval from the U.S. Food and Drug Administration for clinical testing and is expected to be mass-produced and launched in 2021. Formosa News, Stephany Yang, Huang Yutun in Taipei.